For this portion of the lesson, you will be working with the assessment statement, particularly the portion about calculating magnification of drawings and actual size of structures, and this will be done using micrographs or pictures of actual structures. In your next class period, you'll actually be doing this by using a microscope and calculating sizes of different structures under the microscope. For today's lesson, you will need to make sure you have a ruler and a calculator available to you. In addition, you will want to make sure that you have accessed this Google Doc from our Canvas page, and as you're watching this video, you will be taking some notes on this document. You will eventually be putting your work for your calculations down at the bottom here, and that will involve taking pictures of work that you do out on paper and then uploading those pictures to this document. So moving along, I'm going to show you a video, and I would like you just to think about whether or not the video makes you feel really large or really small. Okay, as we're looking at this video here, we are at the level where things are 1 meter in size. So a human is actually a little bit taller than a meter, but it just gives you kind of an idea of size here. So we're going to zoom out, and as I move the bar here, you can see, if you look at the bottom right here, now we're getting larger, things that are larger than a meter. At this point, we're at a kilometer, 10 to the third, or 1,000 meters. Starting to see states. Now we're at 10 to the 6 meters. Starting to see moons and planets. Now we're starting to get quite a ways away from Earth. We'll go back quickly to where we started. Now we'll go the opposite direction. And we're going to structures that are smaller and smaller than a meter. You see cell organelles, bacteria, chromosomes, smallest thing visible to an optical microscope, HIV, hepatitis, viruses, DNA, cell membranes. They're getting smaller and smaller, and we could continue going on this. But that is just to give you an idea of some units and some sizes of different things. As we talk about measurements and units today, we need to understand the measurements that we'll be using. And so we'll use the meter as our standard measurement. 1 meter contains 10 to the third, or 1,000 millimeters. It will contain 10 to the 6th micrometers, or microns, these are sometimes called. And it will contain 10 to the 9th nanometers. So something that's a nanometer in size is incredibly tiny. If you picture a meter stick and think about 10 to the 9th of those crammed in there, that tells you how tiny a nanometer would be. So just to start thinking about sizes of living structures, I would like you to put numbers 1 through 7 on your note sheet in order of increasing size. So that would be number 1 would be the smallest structure, number 7 would be the largest structure. Here's a diagram that might help you. You can go ahead and pause the video to complete this. This gives you the actual ranking. So atoms, molecules, membranes, viruses, bacteria or prokaryotic cells, organelles like the nucleus within a eukaryotic cell, and then finally the largest should have been eukaryotic cells. So today we're going to figure out how can we actually determine the size of some of these different structures. So how do you think you could use the information in these pictures to determine cell size? Pause the video for a moment and discuss that with your partner. Well, in order to do that, we're actually going to use this formula here, magnification equals size of the image over the actual size of the specimen. So today you're actually going to be using rulers and calculators to determine this. In lab next class period, you'll actually be using microscopes, and you'll be using this structure that's within the microscope for identifying sizes of cells. So we're going to actually practice with this particular picture over here. This happens to be the human egg cell. Our goal is going to be to figure out the magnification of this image. So the problem tells us we want to know magnification. So I know I'm solving for magnification, so I want the size of the image over the actual size of the specimen. Well, to solve this, I'm going to look right down here at the scale bar. Here's where I have an image and an actual size. That scale bar is telling me that the actual size that space represents is 40 micrometers. So I'm going to put 40 micrometers on the bottom. And to figure out the actual size of the specimen, I literally need to take a ruler and measure the distance here, which I did earlier, and the distance of that scale bar, or the length of that scale bar, is 18.5 millimeters. Well, we know that we have a problem here because we can't have two different units on the top of a fraction, so I need to convert my millimeters to micrometers by multiplying by 1,000, so I now have 18,500 micrometers over 40 micrometers. And if I divide those, if I put that into my calculator, I end up with the magnification being 462.5, or it's magnified that many times. So this is magnified quite a bit over real life. This is a problem that was on your pretest, and if you got to it, you might not have been sure how to solve it, but the question is asking for us to determine the width of the chloroplast. So this entire structure here is the chloroplast. I want to know what the width is right here where this double arrowed line is. So there's a variety of different ways I could solve this. I could set up the problem like I did the previous one, where I use magnification equals image over actual size. And I could use the scale bar down here, and I measured that scale bar, and it's 1.6 centimeters. So I could put 1.6 centimeters on top and 1 on the bottom and calculate the magnification. Then I could measure the line here and do a similar calculation using my value for magnification. However, since I know both are going to be set equal to the same thing, I can also just set up a proportion. So I can take my image size, 1 micrometer over 1.6 centimeters, equals what I'm trying to figure out here over my 6.3 centimeters. Now this time it's okay to have different units as long as I line them up on my side of the proportion. And if I solve for x, so I cross multiply, I get 1.6x equals 6.3, divide both sides by 1.6, x ends up equaling 3.9 micrometers. Okay, the centimeters would cancel as I do the math here. So together with your partner, I would like you just to talk through the thinking and the process that you would use to solve these following two problems, and then type that thinking and process into your note sheet. You do not need to actually solve the problem. So how would you calculate the magnification of this image, and how would you calculate the width of the cell as marked by the pink line? So pause the video and complete that portion of the note sheet. The next task that you are going to do with your partner is you are going to work through these problems that are on this sheet. You should already have a paper copy of this sheet. If for some reason you don't, you can access it on Canvas. It just makes it a little bit trickier to do the measurements if you don't have a paper copy in front of you. Notice in bright pink up here it says show your work for all calculations. You will be writing your work, handwriting your work, and then taking pictures of it and uploading it into that note sheet that I gave you. So I'm going to go through the first question with you, and we'll see how we can do, and then you will work on the others together. So the first question here says, determine the magnification of the string of thiomargarita cells in figure 8, that's over here, if the scale bar represents 0.2 millimeters. So I am trying to figure out magnification. So I can use my formula up here. Magnification equals the size of the image over the actual size of the specimen. So I'm solving for M. I need the size of the image, and I need the actual size of the specimen. Well, it tells me the actual size is 0.2 millimeters. So what I need to do is I need to measure, that's the scale bar, I need to measure the scale bar itself and see what my top number should be. So if I bring my ruler over close here, as I look at this from 12 to a little over 14, remembering that 12 to 13 is 1 centimeter or 10 millimeters, looks like about 21.5 millimeters to me. So I'm going to put 21.5 millimeters on top. My units are the same, so I'm good there. So my next task is to get out my calculator, and I'm going to take 21.5 divided by 0.2, and I end up with my answer of 107.5. So this image has been magnified 107.5 times. Okay, so that would be my work for 1A. For 1B, it says determine the width of the string of cells. Well, I think it's easiest to determine that by first of all drawing a line, so I know what I'm measuring. So I'm going to draw a line right across here. And I used a ruler to do that, although it didn't end up looking real straight. But then I'm going to grab my ruler and I'm going to bring it over here and measure. When I measure the diameter or the width of this string of cells, I get about 30 millimeters. So I'm going to use the same formula that I had up top, only this time I know the magnification because I just calculated it. Okay, and so I'm going to plug that in here. I know my measurement was 30 millimeters, and now I can solve for x. And by solving for x, then I can get the width of that string of cells, and my units should be the units that I measured in, so my final units will be in millimeters. Okay, so that is how you and your partner should work through the rest of this activity, putting answers in the note sheet that I gave you and taking pictures of the work that you do and entering those into that same worksheet also, and then submitting the entire thing via Canvas. Please feel free to reach out and ask questions if you have them.